You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech Health Podcast, and I have Dr. Berg. She's the founder of a company called Alto Predict. So, Ellen, thanks for coming. How are you doing?
Great. How are you?
Good. Tell me about Alto Predict. What's the premise of the company?
Yeah, so Alto Predict is a data science company founded to promote adoption of non-animal alternatives for product safety testing. It's focused specifically on human in vitro technologies.
Okay. What are human in vitro technologies?
Great question. Primary cell-based assays, organs on a chip, organoids, bioprinted tissues, microphysiological systems, stem cell-based assays, all in vitro human-based assays. The idea is to use these in place of animals for testing for safety.
Yeah, I just have I'm not on the inside of this. I just have the general feeling that it seems like less animals are being used for testing, and I think the public, it's just my guess, the public feels like everything's okay now and animals aren't used very much, or, if they are used, it's just laboratory mice, which I guess not a lot of people seem to care about. But is that true, or what do you see?
No. No, it's not true. That's the problem.
That's all I wanted to know. It's an urban myth.
Yeah. So the reality is, despite all of these innovative advances in these methods, and so many different platforms that have been developed over the last 10, 15 years, there's a tremendous lag in adoption by product developers to use these systems. The reason is, multiple reasons. There are many options, right? Many different platforms, crowded market, many different stakeholders are involved in acceptance of these assays and replacement of animals. And the data from in vitro assays, it's complicated. The analysis isn't trivial, and the validation data from these new platforms is either very limited or very hard to access. We often see, publications where the data are in supplemental table 4 in an Excel table. And so it's not easy for the people who need to agree to use these to gain confidence in their use. So our idea is to build a platform that provides easy access to this validation data. So we're going to collect it and integrate it, along with building specialized analytics and data visualization tools, sort of bring business intelligence analysis to this complicated scientific data.
All right. Well, another misnomer or misconception. In vitro, I think of in vitro fertilization. That doesn't mean in the body. I think in vivo is inside the human body or inside an organism's body. But what's in vitro? That's
correct.
Outside in a dish.
Outside in a dish. Exactly. And the the challenge is, how do you model in vivo biology, complicated in vivo biology in vitro? But the technologies are developing. We're able to culture many different cell types now, and from stem cells induce the differentiation into things like neurons that you can't really get human donors to volunteer their neurons for testing.
Right.
So one thing that hasn't been appreciated is that, toxicology, safety testing is just a very conservative discipline. And people are very cautious to adopt new things unless they're really sure it's going to be better. So we need to apply now data science to pull together all the data to evaluate these platforms and test them really well and determine whether or not they're predictive in whatever application product developers need.
Well, what's the assumption? That animal models are predictive, or they're just some points of data along the path towards a clinical trial, or what's the common perception right now?
Yeah, it's legacy. The concept many years ago was, well, if something is toxic in an animal, it's more likely to be toxic in people. Absolutely true. However, as we have learned more and more in depth about the mechanisms of toxicity and the drug mechanisms, we now appreciate how different animals are. Even their, gene sequences are slightly different enough to make it us unable to predict effect, from animal testing. And the other reason is a lot of the in vivo animal data is in silos within pharmaceutical or consumer products companies, and nobody has access to that. So it's been hidden, if you will. The lack of predictivity of animal testing has really been hidden. It was the best we had at the time, but as we learn more about the details, the mechanisms of human biology, it's quite remarkable how different people are from animals, and it makes us realize why drugs still fail 30% of the time for safety in people.
Well, I guess we don't know if these other methods are the best available because, like you said, data science has to be done on them to evaluate that, right?
Yes, and actually that's where I come in. I've spent the last 15 plus years developing a human in vitro platform using human primary cells to model different aspects of tissue biology, and we've successfully discovered a number of novel mechanisms of toxicity that we have used and developed as commercial assays, and are helping people in drug discovery select safer molecules, and we've shown predictivity of these assays. So that's where I come in, because I have had the experience of, once we identify assays and test them and do the work to show how well they predict human outcomes, we just can do the same thing for many other different platforms.
All right. So what is, I don't know if the word typical is right, but what's an example of your platform? What constitutes it, and what have you tested it on, for example?
Yeah, so right now we're working on our beta version of the platform, where we have a partnership with Collaborative Drug Discovery, which is a cheminformatics biological database, commercial database, to showcase collecting various in vitro assays that are relevant to cardiovascular toxicity. And the area we're working on is the tyrosine kinase inhibitors, which is our class of drugs, oncology drugs that are efficacious in oncology, but cause cardiovascular toxicity. And we pulled together a number of different assays that covered different mechanisms of cardiovascular toxicity. And I think the field hasn't really appreciated how many different mechanisms there were, and that this particular class of inhibitors actually hits. Some of them have different mechanisms of cardiovascular toxicity. This is important because if you're going to, yes,
right, okay, let's get some details. So what's the goal of the drugs, and what do you mean by toxicity? What happens to the heart?
So cardiovascular toxicity outcomes in patients would be increased heart attacks, hypertension, so you get increased blood pressure, or you're at risk for thrombosis, stroke, and those sorts of outcomes. And so what we see in the clinical studies with drugs like trametinib or Mekinist, which is a drug that's used in melanoma, is that there's an increased number of patients who take that drug who have cardiovascular these events the number of events are increased. So when we started
the pathways that are commonly thought of now, and what are the new pathways that it looks like you're figuring out?
Yes. So most people for cardiovascular toxicity, they test heart cells. So these are cardiomyocytes, and there's some nice new assays using induced pluripotent stem cell derived cardiomyocytes, and they beat in a dish. So that assay has been used for, people have looked to that assay to predict cardiovascular toxicity. Well, it turned out for trametinib is that it wasn't active in that assay at all, but it was active in a different assay that modeled atherosclerosis type of biology. So that's where the vasculature is inflamed, and that can also result in increased cardiovascular events, for example. And when we looked at a number of these different kinase inhibitors, we found that some of them were active in the cardiomyocyte assay, that's measuring arrhythmia, and then others were active in assays that measured thrombosis potential, and still others like trametinib were active in the assays that modeled this, atherosclerosis or hardening of the arteries type of biology. And so just in our case study that we're working on now, we could see that this would be very, very helpful for our stakeholders to be able to look across different assays and understand where they might be predictive of human outcomes and get comfortable with them. So that's the type of projects they're doing.
Well, how do you know if an assay is predictive or not? If it still has to go on a path of multiple years to see if in actual people, I don't know what phase it is of a clinical trial, for instance, but where it's actually used in people. And then you'd have to go back to the assay and say, it was predicted here, but not in the people.
So we're lucky because we're in these assays, we're testing all approved drugs, or failed drugs. And so you have all of the clinical data from those to show you how predictive your assays are. And that's really it's challenging because most small companies who are developing some of these assays, they'll only test a few drugs, but you need to test a lot to be able to determine how predictive the assays are. So that's what we're trying to do by integrating the data and supporting all of these vendors or people developing these assays to run the data, run the assays that are needed to show that how predictive they are.
Do the failed drug makers want you to do this, and the successful or approved drug makers not want you to do this for fear that it may unmask problems with their drug?
They've gotten over that. The water's under the bridge, right? And for every company who has a failed drug who doesn't really want to look at it anymore, other companies are looking for those opportunities. Well, if I could fix that drug, then we've got something. So in this space, it tends to be less competitive than efficacy. So there's a lot more sharing of results among product developers in the safety testing area. So it's a little bit easier than some other research areas.
So you mentioned, 7, 8 different kinds of assays, organoids, other stuff. What assay are you? I would guess you're focusing on. I know it's condition dependent, I know. But what type of assay are you focusing on? And tell me a little bit about how it works.
Yes. So I'm interested in integrating lots of assays. The platform that I co-led the development of has 60 plus assays with many other endpoints. So I'm used to looking at a lot of different assays for different types of biology. And I'm interested in in particular assays that have been scaled and are shown to be reproducible. So it's anything that and has a reasonable scientific rationale and that are available, especially commercially, so that you can run the reference drugs and generate a large data set from those. I think that's one of the actually more challenging aspects. There are a lot of academic groups who will run an assay, but it's not particularly reproducible. So, you can't make a large database because it's not reproducible from day to day or week to week or whatever. So just by focusing on those that have undergone some optimization and effort to get reproducibility, it's a reasonable number of assays, and we're about the data anyway, so the more data the better.
Well, I would still think that for instance, organoids, maybe a heart organoid is well understood and pretty easy to make, but a kidney one is still impossible to make. So I would think you'd have to focus on maybe a cell type that works to make 8 different kinds of assays for it. So then you could really get a robust sampling. Then you could test, 100 different drugs that affect just the heart. You're just using heart cells, and heart cells are great because you could do 7 different kinds of assays, and they're repeatable and all that stuff. So I would think you would have identified that, right?
Yeah. So the challenge is the more complicated the system, so the like organs on a chip, the fewer, the lower throughput there is. And so it's very difficult to get enough data to even be confident that your system is showing the biology you expect. So there's a challenge with very complicated systems. And we've had more success where we have tried to just model a particular aspect, maybe, a few different cell types, or, not the entire organ, but a particular, set of biological process that captures an aspect. And then by combining these together, that's where you can cover more of the biology.
So this is kind of like the example you gave, the heart. Maybe you can't reproduce a whole heart, but you can reproduce part of it that's responsible for the beating aspect only. And then you look for arrhythmias, from 100-something different drugs. And then maybe you add in another aspect of the heart,
Yeah, yep, exactly. And we've had success in doing that. So that has certainly inspired me to just expand the data that we can put together and try to connect the knowledge that we've gained by testing all of these drugs and experimental compounds in these assay systems. We've learned a lot about how biology works. So it's been very rich and very exciting. And the idea behind what we're doing is to actually make this reference data, validation data, completely open and transparent, because we need that to have our stakeholders be able to have access to the validation data and be comfortable in these assays that for them to bring these on board.
You're not having to give away some of your secret organoid sauce, so tell me about this, right?
Yeah, so it's about, but what people really want is they just want to know, they want to see that it works. They're more interested in that it works. The secret sauce remains with the different vendors who are running the assays or developing new ones.
So what kinds of things have you learned about biology that most folks may not know about, or you didn't know about before you started doing this stuff?
Most people don't appreciate the hierarchical nature of biology. So, you've got a molecule, DNA or a protein or a drug that might interact with a protein inside a cell. That protein actually interacts with other partners, right? Complexes, pathways, and then pathways interact with each other inside cells. Cells interact with each other in tissues. Tissues make up organs, and there's all this communication between organs until you've got the whole system, the whole organism. And for us information that is closer to the clinical outcome, right? Like a biomarker, something that's close to the clinical outcome is more valuable than a gene sequence or knowing whether or not there's a particular gene expression pattern. Genes are, I think of them as the lowest level. It's like the alphabet, right? And you can't build your understanding of a system by looking at the letters. Because there is all of this modularity and hierarchical organization that you cannot tell from knowing and gene sequences or gene expression. So I think
that's a problem because you have to be reductionist in order to be able to do this, but you can't be reductionist because everything is interconnected and has a hierarchy. So what do you do?
So that's where these systems come in, these in vitro systems, because we're using whole cells, right? We haven't taken them apart and isolated the proteins to do biochemical assays, and we then test things in co-culture, so you can start to put cells together, and when you get the higher levels of organization, they just do different things than the individual cells do, and so just trying to do that helps you learn more about how the system is organized. So
what's
I think that's
an example of that? Because this sounds like juicy, really interesting stuff.
Yeah, well, we kind of know this from gene knockouts, right? You take the whole organism and then you knock out the gene. So you've got the whole system intact, working, and that does help you figure out what things do. Let's see, some of the things that we've appreciated is how important tissue context is, right? So a fibroblast in a lung is just very different than a fibroblast that's in the heart. And the reason is that when you put cells together, they talk to each other and they create their own sort of environment that is not predicted by the individual cell types. So we can look at clinical data from a clinical trial, look at the tissues to see what's going on in human clinical, and then you go back to the in vitro systems. You just keep comparing what you see in vitro with what we know is happening in a tissue, and improving our models, slowly and surely.
So do you see what you consider to be emergent properties when you get enough cells together of different cell types?
Emergent property, absolutely. And the other thing is appreciation for how drug target can have a biological function, many biological functions that are quite different depending on the tissue and setting, and just completely remarkable.
Well, you keep teasing me with these ideas, but what are any specifics you can give that won't reveal things you don't want to reveal?
Yeah, sure. So a few years ago, we were working with the Environmental Protection Agency to characterize environmental chemicals across these biological assay systems, and we noticed in these human systems that they were, responsive to polycyclic aryl hydrocarbons. So these are pollution, right? Mm-hmm. The stuff in pollution. And we noticed that, it had particular activity. And the EPA was all very concerned with, pollution-related cardiovascular effects of these molecules. And what we observed was that these molecules were also quite active in immune cells and modulating the immune cells. So immunomodulation, not immunosuppression, but modulating. And it turns out that we were able to find a biology that is now is now being developed for skin drugs. So they exploited this particular biology that we discovered through looking at environmental pollutants to become something positive. So, and I think that's a really good take home message as well, is that you can find a drug or a chemical that has a particular activity in biology that it might be beneficial or it might be detrimental depending on what is the indication and what is the status of the patient. So
all right, so I got a two-part question for you. Since you're like eyes deep in complexity, let's see. So is it true that the percentage of drugs that are approved appears to be declining? And does that seem to, that and your knowledge of biological systems being so complex, integrated, communicative, and have emergent properties, and et cetera, those two combined, does that tell you that looking for a drug to fix a particular biological problem is a fool's game and it needs to be more sophisticated? Or as long as we have better tools to figure this stuff out, we'll be fine?
I think once these tools become more widely adopted, we'll see a big upswing in approved drugs. These haven't been around that long, and actually last year we had pretty good number of approved drugs, although most of them were for very small niche applications. But I think that is pointing the way that going to be moving into personalized medicine for sure. And it's tailoring the drug to the patient, to the necessary biology. Because I think what we're learning from this work in terms of the complexity of disease biology or the variety of disease biology, that our classification of diseases is not refined enough. And I think as we combine what we're learning from all of the health technologies in terms of individual variation and particular type classifications of patients, we'll be able to start matching drugs and drug combinations with the right patient. I think we're at a lull, but I think it's going to explode.
I guess to throw in even more complexity, certain tissues are adjacent to or intermingled with the microbiome.
Yeah.
What do you do in those situations? Can you create assays with the microbiome or without it? Because I would think that would affect function tremendously.
Absolutely. We've actually explored a lot of microbiome-derived products, and they're certainly very bioactive, and it makes so much sense that they can control allowances. I think we don't know a lot still, the diversity and the consequence, because they're very complicated, right? There's all these species, there's what they make, when they're different combinations will be different. And I think, and once we start to get the data from, comparing microbiomes across patients and really analyzing that information, which we're just at the beginning of. I think we don't have a lot of actual information yet. There's certainly some tantalizing reports here and there, but I think that is an area that is going to be super important because we're a product of our environment, and one of our biggest environmental perturbations is our microbiome because it's sitting in our gut and talking to us all the time. And I like to think of that, we're responding to everything in our environment, right? Food, the air, the people we're around, dog, pets. So we are exposed to all of these inputs all of the time.
That's why I'm surprised that, drugs work, because it seems like, I don't know, just like my personal theory is that it seems like, I guess, we were lucky, for instance, with antibiotics. They were like the low-hanging fruit, and perhaps they're all gone or there's very few left. And, what's the next complication we'll have to do or the next sophistication in order to help people with disease? It involves so many things. It's like, I don't know how your brain doesn't melt down trying to figure out what to do and what complexity to have and not have. It's hard.
Yeah, and I think we have a problem with legacy thinking. So Alzheimer's, right? The researchers are so wed to these old theories, and they keep running the same experiments and trying to prove the same, mechanisms, and, they have not worked. And I think it's very challenging to support and promote really novel thinking and novel approaches. And I can tell you from my experience, I had this idea to use human primary cells in vitro to try to model tissue biology. This was 20 years ago. And I thought for sure, and we started putting together some assays, and they were really helping us learn a lot. And I thought, everybody's going to do this. And it turns out that nobody is. They don't. It's interesting, why don't innovations happen or move forward? And that's a very interesting. It's psychology, it's legacy, and it's overcoming a lot of these barriers. And it's very hard to get novel things published. I'm not going to name any names, but I had a very hard time getting a paper published because in the field, it was believed that this particular protein could not be made by a cell type that we had identified it being produced by, and couldn't get the paper published. So yeah, it always comes down to people.
Yeah, and I guess when you have opinion getting in the way of science and fact and evidence, we got a big problem. It's going to make it even harder to figure out treatment.
Yeah, and I think the technologies are getting so complicated that not enough people are experts, and this is a particular problem in data science, in the life sciences, because there aren't a lot of experts. We're not training our biological researchers in really good data science. And so we're missing out on a lot of good work because we just don't have the people to analyze the data.
Well, before we wrap up, any other case studies that, you really liked or you thought were fascinating, or emergent properties that just you were shocked at?
Yeah, that's a great question. I'm shocked all the time. One thing that I love about this work
what are they?
is that we learn new things all the time. I was particularly surprised that antibiotics were very active on normal human cells. And so some of these work not only because they attack the bacteria or, target organism, but because they actually recruit human biology. Another interesting thing is we did some work with material science, and these were implant devices, and they were concerned about a reaction in clinical trials to some of the materials, but not others. And so we were trying to find out what's wrong with this material that it causes this reaction. And it turned out that it was actually the comparative materials were interacting with the human biology and suppressing. So it was the control samples that were active, and how they recruited the cell responses, was just very interesting. So that's one interesting and exciting application of these in vitro technologies is you can test all kinds of things in them, and extracts and natural products and as well as, drugs, any consumer use products. Is
there not a need then for an IRB, since you're not, working with a living creature, but just a living tissue?
No, what you do want to do is to make sure that the cells, if they're taken from, human donors, that they're consented appropriately. So that's something that you need to pay attention to. But with new approaches for growing cells and expanding them, it's pretty easy to get enough cells these days. And of course, through IPS technologies, the stem cell technologies, they're manufacturing buckets of cells. So that's not so much of an issue.
Interesting. Okay. So, what's ahead for the next 6 months to a year, and then maybe 3 to 5 years for your work? What are you working on right now that you're most excited about? What's coming?
Okay, so right now for Alto Predict, we're focused on getting our the portal up and some data visualizations in place, and so that people can explore the data, and our group of collaborators can help us improve that and tailor that. We have many different organizations have offered to donate data and expertise, very interested in this. Obviously, I'm part of some groups that that their goal is to promote the adoption of these methods. So these are American Society of Computational and Cellular Toxicity, ASCCT, Society of Toxicology, groups, governmental organizations here in the U.S., for sure, the EPA, NTP, NIEHS, FDA, and there are a lot of NGOs that are very involved in helping us do this. Physicians Committee for Responsible Medicine, the Humane Society, their Biomed 21 program. So there's a lot of groups who are supporting and helping us put this together. So that's what we're going to be working on in the next 6 to 12 months is getting it going, building the community, getting feedback from research scientists to see how the data visualizations, how useful they are, and then helping different assay developers get access to researchers who will visibility to their platforms and help them commercialize them. So that's what we're doing there for sure.
Okay.
Yep.
So what's the best way for for listeners to maybe get in touch, questions, collaboration, that kind of thing? What are some resources for them?
Yeah, so we have a website, altopredict.com, and occasionally we have a Twitter account. So either there on Twitter or at our website, they can stop in. There'll be certainly a lot more news coming in the next few months through the website, but that's probably the best place.
Okay. Well, very good. Well, I appreciate you coming, and it sounds like you have 10,000 lifetimes of work ahead of you, but it's exciting stuff.
It's all fun.
Very good.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.